This video is a short overview of our CPS Frontier project titled BioCPS for Engineering Living Cells, which is a collaboration among Boston University, the University of Pennsylvania, and MIT. Our goal in this project is to synthesize global behaviors and populations of engineered cells. On the right, we have some examples of global patterns. The top figure was produced by simulating a network of locally interacting engineered bacterial cells, while the other two are preliminary experimental results in E. coli and human-induced pluripotent stem cells, both from the Weiss lab at MIT. Pattern formation is an important problem that impacts various areas such as amorphous computing, tissue engineering, and drug development, and can also lead to a better understanding of morphogenesis and developmental biology. This project is enabled by two emerging technologies. We use synthetic biology to equip cells with sensing, communication, and decision-making capabilities. Here we show an example of a genetic circuit designed and built in the Weiss lab at MIT that allows for long-range communication using a diffusible molecule called fluoretin and short-range communication through direct contact using a protein that protrudes outside the cell called delta. The decision-making is implemented by the gene regulation interactions, which can be activations, shown in green, and inhibitions, shown in red. We use micron-scale mobile robotics to optimize the formation of patterns by affecting communication and/or by moving cells and beads. We are developing a robotic assistant that consists of micro-robots that can move under the action of a controlled magnetic field. In collaboration with SRI International, we are investigating different types of actuations, including magnetic levitation. The micro-robots combine organic and inorganic substrates. They can interact with cells in culture through direct contact or through diffusible molecules. One of the main challenges in this project is to determine time instance and locations of robot interventions to optimize the formation of global patterns. Other control points in this system are times and locations of light patterning that can trigger optogenetic circuits in the engineered cells. Desired patterns are specified through a friendly user interface. The pattern classifiers are learned as formulas in a novel logic called tree spatial superposition logic. The main advantage of this approach is that given a learned classifier and an image, we can quantify how far the image is from desired patterns, and this distance can be used to map a pattern synthesis problem to an optimization problem. Given a pattern specifier, the next step is to synthesize individual gene circuits and corresponding local interactions leading to the formation of desired global patterns. We use a novel approach called temporal logic inference. In short, using tools from machine learning and formal methods, we map global pattern classifiers to temporal logic properties that need to be satisfied locally by each circuit. Here is an example of such a temporal logic property, which is a formula in signal temporal logic, and the bottom of the slide shows how this formula can be read in plain English. The next step is to construct gene circuits satisfying local requirements as just explained. We are developing a fully automatic workflow that takes as inputs such specs and automatically generates code to control robots in the wet lab. This framework, which is called biodesign automation, consists of 4 steps: specify, design, build, test. In the specify step, we combine the desired circuit specification derived from pattern synthesis with structural specifications, which model compatibility of DNA composition, to populate an exhaustive list of candidate circuits. Each candidate circuit is composed of modules, which can be built in the wet lab using characterized parts such as promoter, ribosome binding site, gene, or terminator. These candidate circuits form our exhaustive design space, which will be used in the design step. The design step focuses on generating a structural topology that can realize the functional specification of the genetic network. This can be done by exploring the design space generated in the previous step. Consider Cello, short for cell logic, which is a genetic logic compiler. This tool uses hardware description language like Verilog to define the behavior of a genetic network and synthesizes a genetic circuit whose simulated behavior can best realize the function of the network. The generated designs are part of the exhaustive design space, which is then built in the next step. The build step automates the planning and assembly protocol required to build an in vitro genetic circuit from an in silico structural topology. Tools like Puppeteer can be used to schedule project plans, track resources used in the project, access daily protocols via calendars, and track project progress in real time. It can also generate robot scripts that can be executed by liquid handling robots. In the test step, tools like Phoenix simulate the behavior of gene circuits and compare the simulation results with experimental characterization to learn model parameters. These model parameters can then be used to generate stochastic models that are checked against the original specification using statistical model checking. So far, we discussed how to use synthetic biology and biodesign automation to build synthetic circuits from existing parts in an effort to achieve global patterns. There are 3 other ways in which we can intervene in the effort to maximize the formation of patterns. We can set up specific initial conditions, such as inducer concentrations, in our robotic setup. We can modify parameters, such as promoter strength and ribosome binding site activity, in the wet lab. And, most importantly, we can determine locations and timings of robot interventions, which, as explained before, can be direct contact with cells or delivery of diffusible molecules. These are all variables in an optimization problem in which the goal is to maximize formation of global patterns. Some of this computation is performed offline before growing the culture, while some is performed online to take advantage of feedback information that we get during growth. We apply these techniques to 3 different systems, one of which is the bacteria E. coli and 2 of which are mammalian, specifically Chinese hamster ovarian cells and human-induced pluripotent stem cells. In particular, we are interested in differentiation of stem cells into liver and brain cells. The movie on the right, which was recently produced in the Weiss lab, shows such a differentiation process, where we can observe endoderm formation in the first few days of cell culture. We would first like to thank the NSF for funding this project. We thank the teams at Boston University, the University of Pennsylvania, MIT, and all of our collaborators, including SRI International. Above all, we thank you for watching.